Thanks, Chuck. All right, it's always exciting for me. They only let me do this once a year to talk to people about instruments. I'm an analytical chemist originally, and I've only got 30 minutes, so hang on. Then the other side of that is I apologize. Most of you probably won't need to know most of what's in this session. If you're submitting samples to the core lab, you're going to be running. We're going to be managing this side almost entirely. So if you want, it's transparent. You don't need to know any of this in detail. But that said, I think it's actually a really good idea for you to know a lot about this because what I want you to be able to do is to be able to read the methods section of a paper and understand at least the basics of what's going on. If you're going to be working in the metabolomics field, that's really helpful to have that basis of understanding. And in defense, there's a couple of you, we look at those forms, just to reiterate what Chuck said, and two of you actually checked instrumentation as the most important part, which is the first time that's ever happened of this talk. So thanks, whoever said that. They won't tell me to take this out entirely, but in general, this is the least liked session because no one needs to know about instruments. Anyway, okay, all of that said, enough apologies. We're going to describe the role of instrumentation and explain why we might pick a particular instrument for a particular metabolomics analysis, and that's what I already said about wanting to know the key terms so you can read a paper. Where are we? We are, let's see here, maybe we're here. There, that's where we are, separation, detection, instrumentation. That's what I'll talk about. The outline, so what tools do you need for metabolomics? What makes an instrument good for metabolomics? NMR versus Mass Spec. I'm not going to talk about NMR. For those of you that don't know, we're a very heavily MS-based facility. If you need NMR, or if we have the sense that your project would be better suited to NMR, we can put you in contact with it. Well, we will aid you in getting your samples analyzed by NMR, probably here, and if not, there are other centers that have other expertise in NMR among the MRC squareds. Okay, and then we'll talk about hyphenated techniques. You've heard of LCMS, GCMs. Why do we need to use a technique with a hyphen in the middle? I'll tell you what that means. Chromatography, we'll look at methods just so you're familiar with it if you're not already. Mass spectrometry, this is the majority of what makes the technique possible. Actually, really, it's a combination. And then we'll look very briefly at the instrumentation we have here, and then that's it for one more year. Can't talk about it anymore. We have, so the instrumentation and omics science. There's really an intimate connection between instrumentation, analytical methodology, and the development of these omics sciences. Before, to sequence DNA, you did Sanger sequencing. You ran long gels. You carefully looked at positions of different bases, and then you came up with a DNA sequence. It took forever. Thank goodness we've had advances in instrumentation, and then we had shotgun sequencing and next-gen sequencing, which makes incredible advances as far as speed and throughput and accuracy of, maybe not accuracy so much, but speed and throughput of sequencing possible. Same thing happened with proteins. Maybe 2-3 decades ago, we were doing 2D polyacrylamide gel electrophoresis, and then excising, and then that was all you could do at a best, or maybe you were just going to do a Western blot, and that was all we can do. And those are still very useful techniques, but advances in instrumentation, mass spectrometry occurred, and now we have shotgun proteomics, where we can take the entire complement of proteins in a sample, separate it out, and then try to identify as many proteins as possible. And there are limitations to these techniques, certainly, but it's much faster and high throughput than what was done in the past. Metabolites, same thing happens. We have biochemical assays, nothing wrong with them, they still work, but advances in instrumentation have made it possible to measure many compounds simultaneously, and we have untargeted and targeted metabolomics. Okay, so moving forwards. Workflow. What do I need to know about instruments? Do I even need to know where are we in this queue? We're right here in this middle in the proverbial black box. Yes, this is mainly done by the metabolomics core, but I've already told you why we want to know. So today, for the next few minutes, we'll open the black box, see what's inside, and then we can close it. Tools that are needed. We have metabolomics, the definition again, study of all small molecule metabolites present in a cell, tissue, or organism. So we need something that can can handle that. We need something that's sensitive. We need something that's quantitative, because if you can't quantitate what the concentration of metabolites in your sample, then why are we even starting? We need to compare groups. So we need something that's quantitative. We need something that's universal. We don't want an assay that's very specific to a particular metabolite and doesn't let you look at the other metabolites that are there. If not, then why wouldn't you just do a biochemical assay on an ELISA or something? We want it to be capable of detecting many compounds simultaneously. That's what we're saying here. Same sort of thing as universal. So the instruments that can do that to a first approximation are LCMS, GCMS, NMR, maybe CEMS. Maybe there's a few other techniques that are separation-free or even mass spectrometry-free, GC alone. But that is the sort of suite of tools that's used in mass spec or in metabolomics to the vast majority of cases in the present day. Okay, so NMR. I'm not going to talk to you very much about NMR. Kathleen Stringer tomorrow, I believe, is going to spend a little bit, or maybe it's Thursday, I can't remember, is going to spend a little bit more time talking about NMR and its use in metabolomics. NMR does have some definite advantages. It's a near universal detector. Anything with carbon atoms in it or anything with protons or various other nuclei will respond in NMR. Is quantitative without even having to really work very hard. It's reproducible, provides structural information, and it requires very minimal sample preparation. So those are all good things. Its limitations, main limitation is sensitivity and to some extent resolution. You put everything in one spectrum. There's ways to spread it out, but not to the same extent that you can do with an LC or a GC front end. There are efforts, or there have been efforts to couple NMR with chromatography. It's not as easily coupled as Mass Spec is, however. So the thing to take home, and Kathleen will tell you more about that, but NMR and Mass Spec are complementary. They're both useful for metabolomics, and there will be more detail tomorrow. Okay, so here's just a quick comparison. High sensitivity, definitely the nod goes to Mass Spec. Quantitative reproducibility, NMR does it pretty well, very easily. Mass Spec can be very good, and it can also cause problems if you don't know what you're doing. Yes, Mass Spec does analyze more metabolites simultaneously. Structural information, NMR does a little bit better if you have enough sample. If you have a pure sample, it's a lot easier to deduce the structure from NMR. Mass Spec can give you some useful information. NMR is pretty good with matrix effects. Mass Spec is very bad, so we have to manage that carefully. Mass Spec is easily coupled chromatography, NMR not so much. So we're going to talk about Mass Spec the rest of the session. Okay, now hyphenated techniques. What is a hyphenated technique, and why would you want to use it? Well, a hyphenated technique basically just means you're putting two analytical methods together and in a direct coupling manner. It can be offline, but usually it's done online so that they're transparent, and when the first technique is done, the second is done as well. This slide didn't come out too well, but why would you want to do LC? Well, LC is great at separating complex mixtures. You get chromatograms and you see a lot of detail. We'll look at that in a little bit more detail in a few seconds. But it can be difficult to identify components of the chromatograms when you just see, okay, we have abundance and we have some sort of time, and what exactly is that? If you don't really know what's going on, if you haven't carefully run a whole lot of standards and have really good peak capacity, you may not be able to do what you need to do. Mass Spec is great because it determines the mass of molecules. Now, that's a really useful piece of information. Every molecule, every metabolite has a mass, and that is sort of a unique signature. Certainly other things could have a similar mass, and that gets into problems occasionally. But that's a very good first piece of information to get that a few other techniques will give you. No other technique will give you with the resolution of mass spectrometry. And it's useful for identification. But signals from multiple components are difficult to distinguish and can interfere with one another. So if you just said, well, okay, Mass Spec is great, why not just throw our sample on a Mass Spec and see what we get? Well, that's fine, but you get a lot of interference. Oftentimes you get fragmentation, as I'll show you, and you can have a mess that makes it difficult to tell what's what. So what do? We do combined hyphenated techniques. So just a brief block diagram. This is something analytical chemists like to show a lot of how an instrument works at the basic level, and then they go off the deep end and start building crazy instruments. But this is good enough for right now. An LCMS or GCMS system looks roughly like this. We have chromatography on the front end. We have to make our molecules into an ion in the middle after it comes off of the chromatography system, and I'll talk more about that in a minute because a Mass Spec is an ion detector. We have a mass analyzer, which separates things by their mass or does some other fancier tricks. And then we have a data collector, which reads out the data. And so those are the major steps that are involved in a LCMS or GCMS system. So what does that look like in the instruments that we might have sitting here in a lab? In LC, here's a LC stack with various different components. A pump, an auto sampler are the biggest ones. And then we have ionization, which happens in between the LC and the mass spec. Usually there's a tube running between them. This picture is just made to look pretty so that they don't put an actual working setup here. But the source is on the front end of the instrument. It makes ions. Ions go inside the instrument. Magic happens. I'll show you that magic gets analyzed by mass, and then the data collector is here, and it reads out the signal. A GCMS does more or less the same thing, except it's all integrated into a single box-looking thing. Chromatography happens here. It's basically a big oven with the tube inside, and that tube is the column. Ionization happens here in the ion source inside the instrument. Then there's a mass analyzer, same as in the LC. And then there's data collection as well, and that's what the instruments sort of look like. So if you're ever looking at an instrument, now you can say, I know what those parts do. Okay, chromatography. Let's go in a bit more detail. What is chromatography? Most of you probably have some exposure to chromatography from chemical work, or a lot of people do chromatography to purify proteins or what have you as part of biological work. So I doubt this is going to be too surprising. But chromatography is a method for separating and analyzing components of a mixture based on differential affinity of those components for a stationary phase, which absorbs those components, versus a mobile phase, which is liquid and gas. So the components of a chromatography setup in a very basic chromatography column. This is the heart of the instrument, even though it's really a replaceable component. It's what does the actual work. We have a column, which is a tube. Typically, in the context of an LC separation, it's packed with small particles. There are various other things in a GC. This is actually an open tube, and there are no particles. The stationary phase is just on the walls of this narrow tube. But in an LC separation, we have particles. These are like 5 microns in size, maybe a little smaller. And on those particles, there's coated a chemical stationary phase, which has some sort of chemical property. It's either polar, it's nonpolar. And then we have mobile phase, which is liquid, which is flowed through this column by a pump. Then we've got our sample. We've got a mixture of different things. We want to separate it. So what do? We inject that sample onto the front of the column. Here we have the molecules that were in our sample stuck to the front of the column. And then we start flowing mobile phase through the column. And like I said before, things that have a stronger affinity for whatever that chemical coating is on the stationary phase will stick longer. Things that have less of a strong affinity for it will flow through more quickly. And this is what it looks like. So some of your stuff comes out first, and then you get a signal in your detector when that first component elutes, and that's a peak. Then you move forward from there. The next component elutes, you get another peak in your detector, and that's a signal from that. And finally, your final component will elute, and you get another peak. And so that's chromatography in a nutshell. So the output looks like this. We have a plot which is called a chromatogram. It's a plot of intensity versus retention time, or time. And you see we have different intensities. Different intensities reflect the relative concentration of the things that were coming off the column at that period in time. However, it's not quite that simple. There's different response factors. Some things may give rise to a larger signal for a given concentration. So you can't just say, okay, this is 1 molar, and this is a half molar, and this is 0.25 molar. Usually it's not quite that universal. We need to do a calibration for each component. Okay, so the data output, the units of raw metabolomics data are these chromatographic peaks. They're a plot, like I said, of intensity versus retention time. Here we have a simplified chromatogram just showing a single component, more or less. And the thing to remember is that both the peak area, that's the area under the curve if you do an integration using a computer or however you want to do that, or the peak height are proportional to the metabolite, or whatever it is you're measuring, the concentration of that metabolite. So that's how we get our quantitative information out of a chromatogram. Okay, LC and GC. So there's 2 types of separations. What are they? What do they use? So liquid chromatography separates molecules in solution, have to be dissolved. It relies on the chemical interaction between an analyte, which is in our case a metabolite, and a stationary phase, which is a column, and the mobile phase, which is your solvent. Many different separation modes are possible. I believe the next slide will show you a few examples. Gas chromatography separates molecules in the gas phase. Okay, well, that points out something right there. We have to put the molecule in the gas phase first. So if you can't make your metabolite into something that will vaporize before it burns up, then it's not amenable to mass spectrometry. And to do that, I at one point had a slide on derivatization. I think I took that out, but sometimes we have to play chemical tricks to make our metabolites into something that can go into the gas phase. If you took citric acid and you tried to heat it enough to get it in the gas phase, even at several hundred degrees, the first thing that's going to happen to it, rather than just sitting on the bench, is it's going to burn up and turn into carbon dioxide and water. And so instead, we need to chemically derivatize it to make it more amenable to volatilize, and that's what we have to do for a lot of metabolites. So chemical interactions happen between the analyte and the stationary phase, which is a capillary column usually. The carrier gas is inert. It's something like helium or hydrogen. If you can use either GC or LC, usually GC is the simpler, faster, and higher resolution approach. So GC is great. It's just that it's not as universal as LC. Most metabolites from a biological entity are going to be in solution already. Putting them in the gas phase is a little bit of a trick in some cases. So the metabolite must be volatile or made volatile in the process. Okay, so GCMS versus LCMS, another one of these comparison slides. High sensitivity, GCMS and LCMS can both be very good. Quantitative reproducibility, they both are good if you manage it well, but they both can have their problems if you're not carefully. Both can analyze many metabolites simultaneously. Structural information, GCMS gets a little bit of an edge here because the fragmentation pattern is so rich in GCMS, and I'll show you what that means in a little bit. LCMS is pretty good also. Works with volatile compounds. GCMS is great for that. LC can work for some things that are inherently volatile. Some things that are very volatile don't work too well on LCMS. Non-volatile is not so good with GCMS. If you can't make it volatile, it doesn't work. LCMS is fine with that for the most part. Matrix effects concerns, yeah, there are some in GCMS. It's even worse for LCMS. We have to manage these things, and we do as part of our daily routine in the core lab. A variety of different chromatographic separation modes. There are some in GC, but the LC really gets the nod as far as having more things you can do to play with your separation. Okay, modes of liquid chromatography. This is, you could teach, and people do teach a semester-long course on this stuff, and we have one slide. So don't worry that we won't get all of it. This is just to give you terms that you may run into in reading papers. Normal phase, stationary phase is polar. The mobile phase is nonpolar. Works for nonpolar stuff like lipids, but in this case, it's water-free. So that's not really very amenable to metabolomics in general. There's a few exceptions. Reverse phase LC is great. The stationary phase is nonpolar, works best for nonpolar metabolites in an aqueous, read a metabolite, biological entity-friendly type of solvent system, or a mixed aqueous organic. So this is a good, very useful mode of chromatography. Widely used, applicable to metabolomics. Hydrophilic interaction chromatography, we have a lot more in common with normal phase chromatography. The stationary phase is polar, the mobile phase is nonpolar. It works well for polar metabolites, but the advantage is we've modified our stationary phase a little bit so we can leave some water in the mobile phase, and that's really critical for allowing polar metabolites to dissolve. Other modes of liquid chromatography that you may bump into in literature, there's ion exchange, anion exchange, and cation exchange. Anion exchange retains negatively charged stuff. Cation exchange positively charged. Ion pairing reverse phase is a modification of reverse phase, which makes it more amenable to working with stuff that's polar. Some advantages, some disadvantages, makes your mass spec dirty. Size exclusion and gel permeation and filtration separate things basically based on their size. They're relatively low resolution, useful for proteins, less useful for metabolites in general. So that's it for that. Okay, that was fly through of chromatography. Now we're going to fly through mass spectrometry. Let's make sure I'm not running too long. Okay, we're doing all right. So mass spectrometry, how does it work? Mass spectrometry detects any compound that can form an ion, and that's really a critical thing. So we need to take neutral metabolites after they come off of our column from separation, zap them with some sort of method, make them into ions, separate them by mass. Then we have mass-separated ions. That's good, and then we need to detect them. It's sensitive and selective, which means there's high resolution. I'll show you a little bit about that, what that means later. It can help identify metabolites, give structural information, useful for quantitation. We wouldn't have even tried if it wasn't. But sources of interference and matrix effects must be carefully managed. Okay. So the output of mass spectrometry is a mass spectrum. You get a plot of intensity versus mass to charge ratio which effectively is it's the mass of the metabolite that you're measuring divided by the number of charges on it. In metabolomics, our molecules are small enough that they almost always have a single charge. So normally our mass to charge ratio is just the mass of the analyte, usually plus or minus 1 Dalton, because we form ions by taking off or putting on a proton. That's the way that electrospray ionization works in liquid chromatography. I'll show you about that right now. So there's lots and lots of different means of making an ion out of a metabolite or a molecule. This is just one of them that happens to be the most common that we use in the context of liquid chromatography, mass spectrometry. So what we have is originally we have a flow of liquid containing our metabolites after they've hopefully been separated by our column. And we need to get those things into the gas phase. We need to get our metabolites into the gas phase. We need to get rid of all of the liquid that's coming with it. And we need to introduce it into the mass spec. So what do we do? It turns out that if you apply you put a metal capillary here, you apply an electrical potential across the small amount of space between this capillary outlet and the inlet of your mass spectrometer. You can either put the high voltage on the tube, or you put it on a different electrode in another position. And what happens is you impart a lot of charge on the very small region of liquid coming out of this capillary. And so, you make little droplets that come out with all this charge. And instead of just sort of dribbling off, because of all this charge, they form these very tightly spaced small droplets. And you also add some gas to help, sort of form a nebulized spray as this is occurring. And all this charge is very concentrated on these droplets. You've got a lot of drying gas being pumped in here so that these droplets are evaporating down. And what happens is the liquid evaporates and the droplet gets smaller and smaller, and eventually the charge gets so tightly packed that you reach the limit of the amount of charge that can be held in the droplet. And once you go beyond that limit, the droplet fissions into many smaller droplets, and that process continues. You get smaller and smaller droplets with fewer and fewer ions, and eventually you're just left with solvent-free ions floating around in the atmosphere. And these ions then can be pulled inside the mass spectrometer. Charged particles move in an electric field. You use that, and also just gas dynamics. You've got vacuum inside the instrument. You have atmospheric pressure outside. Those ions get drawn inside the instrument, and voila, we have ions inside of our instrument, and we can do something with them. So what do we have to do? We have to analyze them by their mass. And so there's a variety of different mass analyzers that do this job. So we go from a mixture of many different mass to charge ratios ions that are coming off of our chromatography column. Ideally, we've reduced the complexity by chromatography, but we need to separate them by their mass and then detect them. Ions can only be manipulated under vacuum, so all of mass spec is beyond that ion formation is done under vacuum inside the instrument. Many different types of mass analyzers exist. If you're reading literature, you're going to bump into several of these types of things. There's not enough time to go into the details. I'll give you an overview of about 3 of them, but these are different ones that you may run into, and they all do their job a little bit differently. So a quadrupole is the first one we'll talk about. This is not the original mass analyzer, but one that's been around and well understood and used since the 60s or before. Basically what it does, you take all your ions in, and it selectively passes ions with a specific mass at a given time by controlling the radio frequency and direct current voltage applied to a set of 4 poles. And so you put ions in, you've got these voltages, and some ions make a stable oscillating trajectory as they pass through. Some of them are ejected out because they don't hold a stable trajectory, and you can alter these voltages to change the ion that has a stable trajectory. And based on your knowledge of those voltages and what you detect over here, you can say, okay, when I was holding my voltages that should have passed mass to charge 191, I see a signal, so I have mass to charge 191. This is a low mass accuracy. We can distinguish things that are about 1 Dalton apart, which might sound pretty good, but there's instruments that can do a lot better. It's good for quantitation, and it's actually not very expensive. So this is a good, reliable, robust type of mass analyzer. The second type to talk about briefly is a time-of-flight mass analyzer. In this case, what we do is we basically pulse all of our ions in. This is the simplest one conceptually to understand. We give them all the same kinetic energy, and things that are smaller move faster because you gave them the same amount of kinetic energy. Their velocity is higher. So you put them through a field-free drift tube. You just let them zing along here. And then when you got a very accurate stopwatch and a detector at the end, and you say, okay, I saw an ion that arrived at this time. That means I have a low mass ion. You wait a little longer. The high mass ion hits it, and then you get, okay, I have a higher mass ion. Of course, you're not doing this. The instrument does it for you. It takes a few microseconds for these things to pass through, and you do that thousands of times a second to make a single spectrum. So the advantages of this, high mass accuracy. You wouldn't think it, but it actually gives you resolution that can be down to 0.001 Daltons if you've got a really good instrument. A better likelihood of identifying unknowns, and it reduces noise from the background, but it is a little more expensive and requires careful attention to the calibration. One more type, because you'll probably bump into it if you're reading the literature, which is sort of the newest type that's out there and commonly used for metabolomics, is called an orbitrap, introduced in 2005 by Thermo Fisher Scientific. Detects ions based on signals that are induced as they rotate around a central trap electrode based on some pretty fancy voltages and physics that go on with that I don't fully understand myself. It offers even higher resolution than TOF. This is a comparison of a sort of middle-of-the-road TOF with a full-width half maximum resolution of 10,000, and what you get if you have a resolution of 100,000. So you might, in rare occasions, have actually two components that would be not distinguished by the TOF, but could be distinguished by the orbitrap. So that's great. In most cases, it doesn't actually help that much. The mass accuracy, the ability to distinguish right where the center of that mass is only marginally better. It's more expensive, so you can have fewer instruments if you're going to buy them. Has more working parts. Potentially, I always find that the more working parts an instrument has, the more likely it is to break. Okay, very quickly, make sure I'm starting to run over a little bit. Tandem Mass Spec. How does this work? So you've probably heard of MSMS. And I mentioned yesterday that sometimes if you're trying to identify a molecule that you don't know, or if you're trying to remove interference from the background, it's not enough to just do a single stage of mass spectrometry. It's very useful to do it, but sometimes the next step is to do tandem mass spec. So we've got all these ions in. We could just separate them by mass and then detect them, and that would be what I've shown you thus far. But what if we wanted more information? Well, what we can do is we can pick one or even multiple of those ions going in, and then we can have those ions collide with some other molecules floating around. And when a fast-moving ion hits another molecule, in this case it's usually a neutral gas molecule, it might break. And it might break in a way that you wouldn't really think like a chemical reaction could happen. That's a lot of energy, and there's nothing else floating around. There's no liquids in solution. So you can get some pretty crazy chemical reactions. You can break many, many different bonds and form all sorts of different interesting fragments out of the molecule that went in. And those fragments all now have different masses. So what do? We do another stage of mass spectrometry right here, and we select a particular product ion, or we look at all of those different product ions and measure their mass, and we have now more useful information. So the fragments that are produced by this tandem mass spec process are useful both for metabolite identification and also for improving selectivity. The odds that you have a metabolite that have two metabolites that have exactly the same mass might not be all that low, but the odds that metabolite might have exactly the same mass initially and then fragment to give exactly the same fragment ions is somewhat lower. So that can help offer selectivity in trying to distinguish metabolites. So this is good. It can actually also help out for quantitation by removing chemical noise. But when you do it that way in say, a triple quad instrument, there's a limited number of metabolites that you can monitor simultaneously. Okay, so types of tandem MS instruments. This is the last instrument slide, I believe, other than just showing you some pictures. We have a triple quad, a tandem quadrupole instrument, and so this instrument is really designed to do MSMS in a way that is quantitative for targeted types of experiments. And so you have one quadrupole that, as I showed you before, selects your ions, a second quadrupole, which is actually really usually a hexapole, which means just 6 poles instead of 4, where the collision happens. So you do your first stage of MS, you select your precursor ion, you fragment it here, and then you select your product ion, sometimes called a parent and a daughter ion. And then you can either select your ions or scan through them all, and you've got a detector here. Very good for doing quantitation, limited because it's got 2 quadrupoles to unit mass resolution on both of the first instruments. Another Tandem MS instrument is a QTOF. We have several of these here, and we do a lot of our untargeted work on these. We have a quadrupole, which allows you to select ions, a collision cell, pretty much the same as a triple quad, but then instead of another quadrupole, we put a TOF at the back end of it. And so that's great because it gives you higher resolution of those secondary ions. Not as good in some ways for quantitation because of things that we don't have enough time to explain, but it's great for scanning through and doing identification work. And then the other type that has some prevalence is a LTQ Orbitrap. You can also have a quadrupole Orbitrap. So this basically has the same rough sort of workflow as a QTOF. We put a mass analyzer on the front end. If you have a linear ion trap, you can do a few more fancy things with your ions initially. And then we have a C-trap, which basically just pulses your ions in the Orbitrap. The Orbitrap basically does the same thing as TOF. It gives you a high-resolution readout of your ions. And so these are 3 types of instruments that you may bump into. We have triple quads. We have QTOFs. We don't have an LTQ Orbitrap in our core. We thought that the throughput advantage of a QTOF and the fact that you can buy more of them for the same amount of money leads us to in this direction. And we also have just more of them. There are occasions when this is handy when you're doing identification work, but for throughput advantages and for the fact that you can really do pretty well with a QTOF, we haven't yet purchased one of these. Okay, so instrumentation in the MRC squared. We've got the instruments that you need to get your samples run. We have, and this may not be fully up to date, we have 5 QTOF, 4 triple quad, 1 TOF LCMS. We have 2 GCMS systems, 2 GC flame-induced dissociation systems. And then we have automated sampling, handling, and robotics when we're doing really large throughputs. We have additional sample prep and instrument support equipment. So we have the instruments to run your samples. If anyone's really interested in this stuff, I think you'll get a chance to see the lab tomorrow or come and talk to me afterwards. And I believe that is it, except for the summary. So metabolomics, like other omics sciences, based on the capabilities of instrumentation. NMR and Mass Spec are complementary. Combining separations and Mass Spec is very useful, helps manage complex samples, which we always have in metabolomics. And the core lab uses multiple types of instruments, and hopefully I've explained a little bit about what we do at this breakneck speed presentation. So that is it. I don't have too much time to keep us on time for questions, but I'm happy to take a couple if you've got some. Yes?
Can you just explain what that term matrix effects means?
Sure. So matrix effects are anything that prevents you from doing quantitation on your metabolite by affecting causing different response of your instrument to a given concentration of metabolite in different samples or in different sample types or anything like that. So say we had citric acid in 2 samples, right? And we had a peak area of 500. And normally, if you look at a calibration curve, you could find, okay, 500 corresponds to a concentration of 1 micromolar, let's say. But matrix effects would be something where in fact, that signal of 500 did not actually mean a concentration of 1 micromolar in both samples. It might mean that you would get 1 micromolar, but the true concentration would actually, in one sample, and the true concentration in the other sample would actually be 0.8 micromolar. So it's some characteristic of your sample that induces a difference in the instrument response to the quantity of metabolite that you have. And so how do we manage that? Well, we use standards to do that. We put standards not just in a calibration curve that's run separately from your samples where you don't have any means of managing that. We put standards right in with your sample that assess the magnitude of that matrix effect, that we know exactly how much is in there. We put in 1 micromolar, say 13C citrate, and we see that, oh, in this sample we get a signal of 500 for that 1 micromolar, and in the other sample we get a signal of 450. So we need to adjust for that by increasing our effectively the slope of our calibration curve for that one sample to compensate for that. So it's a bit of a tricky sort of thing. Basically, it's anything that affects your quantitation negatively, and you have to manage it effectively. And we have methods for doing that with Mass Spec. I know that was sort of a long answer to your question. I'm happy to give you more afterwards if you'd like. Other questions? Yes. Right. Well, so metabolites are so chemical fragments, meaning chemical fragments that are present in your initial sample are the things that are, chemically. So all fragments of a metabolite generated in the process of doing LCMS are effectively artificial, right? They weren't in your solution to begin with because they were generated by the collision-induced dissociation, or even if you weren't intending to do it earlier on in your instrumental analysis method. So in that sense, all fragments are artifactual, are not what was in your original sample, right? They can be very useful because they can help you figure out the structural information. But yes, definitely, in the process of doing the analysis, we look at those. We look at those, or the computer, depending on whether we're doing targeted or untargeted. We look at those fragments and say, does this look like something that's coming that is the intact molecular weight of a known metabolite? Or is it more likely that it's coming right along with another peak that is that intact molecular weight, but it's smaller or bigger, meaning an adduct, smaller meaning a fragment, bigger meaning an adduct, and I can tell you more about what that means. And if so, then let's lump that in with the main metabolite so it doesn't get reported as another feature. So with targeted, you generally don't have to worry about that. We know what signal to look for. It doesn't matter what fragments are being formed. Usually the proportion of fragmented to unfragmented metabolites are formed very consistently in mass spec, so we can just pick one and quantitate on that. In the untargeted, it's more challenging. Yes, we get a lot of that signal. For the knowns, we do for the things that we have a library match, we have a pretty good idea that yes, we're looking at the real peak or the most abundant fragment that's best to quantitate off of. For everything else, we use the computational tools that are out there, and then really sorting out the rest of it requires some more good manual work. And when you get to that point in an experiment, the core lab can help you do that, and will help you do that. And we'll give you our best effort, and we're developing tools right now. This is sort of an unsolved problem in metabolomics, not a problem that's unmanageable. It still all has useful data, but a problem that's really of interest to the field to get that data set condensed down to just authentic metabolites. Okay, we got a few more questions. We're going to have to probably keep it moving relatively soon. I don't know. How about this? Come see me after the session. We'll take all of those questions afterwards. Right now, I'd probably better move on in the interest of keeping things on time. So our next speaker is Maureen. Where is Maureen? There she is, over there. Yeah. And